Okay, I think we're ready to go. So today I would like to introduce George Davey Smith, who I met about a year ago in Bristol at a meeting he organized to celebrate Gregor Mendel's 200th birthday. And you can see him cutting the cake, the birthday cake there. The talk he's giving today is one of a series of seminars that George arranged to be given out throughout this celebratory year by researchers with overt links to Mendel, including perspectives from the biological sciences, the history of science, and social sciences. So you can hear these talks if you go to YouTube and you search for Mendel 200 conference. Okay, so George gained his BA from University of Oxford, his degrees in medicine from the University of Cambridge, an MSc from the London School of Hygiene and Tropical Medicine, and a DSc from Queen's College, Oxford. George is a clinical epidemiologist and director of the MSc Integrative Epidemiology Unit at the University of Bristol, and has been a highly cited researcher for every year for the past decade at least, with over 2,000 publications in Web of Science. George is very interested in the relationship between cause and effect in public health. For example, he's used Mendelian randomization analysis to show that, contrary to popularly held views, alcohol consumption has an adverse effect on cardiovascular disease. George has also made many contributions to research on the effectiveness and ineffectiveness of healthcare and health policy. So today, among many other things, George will be talking about randomness and inheritance. That's to say, random allocation of parental alleles to gametes, and the way this randomness can be used to assess causal associations and data. We look forward to your talk, George. Please welcome me. Welcome, George.
Very much. Thanks very much indeed for the invitation. I've had a fascinating morning going around the centre. I've never been here before, and I'm looking forward to more of the same in the afternoon. So, as Julie said, I'm going to talk about randomness in Mendelian processes, and I'm going to start by differentiating the notion of mere chance and pure chance in Mendel's own work. So by mere chance, the term which Mendel used a few times in his paper, what he meant by that was what we now call sampling variation. And he started off the paper by saying that all the previous studies hadn't been big enough. That's basically what he said. And you needed larger studies that could definitely ascertain statistical relations. And towards the end of his paper, he said that the true ratios of the numbers could only be ascertained by an average deduced from the sum of as many single values as possible. The greater the number, the more merely chance effects eliminated. So this was mere chance. Sampling variation is just something which exists and you can deal with by increasing your sample size. Mendel wouldn't have agreed with one of his current acolytes, Alan Franklin, who last year, for Mendel's bicentenary year, wrote a paper called "Want Should Have Been Enough: Mendel's Experiments in Plant Hybridization." Mendel said towards the end of his paper that the validity of the law formulated for peas requires still to be confirmed, and a repetition of the more important experiments is consequently much to be desired. So Mendel certainly thought replication should go ahead. Now, pure chance is more interesting. So Mendel again used pure chance, this term, a few times, sometimes in almost identical sentences. So on a couple of occasions, the virtually identical sentence appeared that it remains therefore purely a matter of chance, which of the two sorts of pollen will become united with each separate egg cell. And I'm suggesting that when he was talking about pure chance, he was talking about what we might call a randomness, true randomness. Now, I'm not, you'll be pleased to know, going into a philosophical discussion about what randomness is. I'll go with what I think Mendel thought and others following him, that you could basically say a process was random if all possible combinations were seen. And we know that Mendel was taught combinatorial mathematics in his rather extended student period. And so he said, you know, it's therefore purely a matter of chance. But according, however, to the law of probability, it will always happen on the average of many cases that each pollen form A and big A and little a will unite equally, often with each egg cell from form big A and little a. So that all forms of combination would be seen in the ratios and numbers expected from that analysis. Later on, he talked about how the behavior of each pair of differentiating characters in hybrid union is independent of the other differences. So this was what would later be termed, you know, the law of independent assortment. So sorry, this would be a segregation. So you'd get the number of eggs and pollen cells would be balanced, so 50-50 in the hybrids. So this was the basis of the random processes in what came to be called Mendel's laws. Of course, Mendel advanced only one law where he actually used the term law in his paper. There were about 7 law-like statements, and Mendel's only law himself was the law of combination of differing traits according to which hybrid development proceeds, thus finds its basis and explanation in the proven statement that hybrids produce germinal and pollen cells that correspond in equal numbers to all the constant forms resulting from the combination of traits united throughout fertilization. So through fertilization. So if you ever refer to Mendel's law, this was what he actually used the term law to describe. Obviously, it's not now what is taught of as Mendel's law. So another aspect of chance in Mendel's life, or in Mendel's afterlife, if you like, is that it may be chance. This is why we've all heard of Mendel. We all know that there were, you know, the 3 rediscoverers of Mendel that all remarkably came across Mendel's paper apparently at the same time, and then wrote their papers. And we also know that this is totally mythical. No one seriously considers Cermak a true rediscoverer. And the case of Carl Currens and Hugo de Vries is interesting. Currens wrote a paper in 1899, so that is the year before the rediscovery year, which just in its last couple of sentences, it just mentioned the name Mendel. He didn't provide the reference. He just said that something that what he was looking at had been these things had been investigated by Mendel. That paper appeared late in 1899. Hugo de Vries, early in 1900, produced a paper where he talked about Mendelian ratios, but didn't mention Mendel. So that came out. This was presented as de Vries's discovery. He presented this as his own discovery. Currens, who was then working on his paper, saw de Vries's first report and then very quickly started writing a paper in which he titled Mendel. He put Mendel's name in the title of the paper. He referred to it as Mendel's law, and he wrote this. In his paper, he said, you know, I did these experiments, and then I was surprised to discover that, you know, that Mendel had done this before. But he gave priority to Mendel. But giving priority to Mendel might very well have been a response to seeing de Vries's priority claim where de Vries didn't do this, and then de Vries, knowing that, probably seeing Currens' 1899 paper, then at least referred to Mendel in his longer version of his report. And we know that both of them actually knew of Mendel's paper way back. At least de Vries later said he'd seen it from the reference from Liberty Hyde Bailey, who I'll come on to, and Currens documented had seen the paper from 1896. So none of them were true sort of rediscoverers in 1900. And who knows that if they hadn't all been working at the same time, and there hadn't been this priority dispute, who knows to what extent they would have referenced Mendel, or certainly probably Currens wouldn't have put Mendel's name in the title of his paper. So maybe it's because those things came together that Mendel then was given the priority, rather than being just a sort of a reference that was then given in a normal way, but was given this sort of massive billing. Of course, there was a later candidate for rediscoverer, William Jasper Spillman, who published a paper in 1901, which was he's a really interesting character, Spillman, a very positive figure in a lot of ways, saying that he hadn't seen the work of others, which is possibly plausible, and he might be the only person who actually had others put up a plaque for him saying that he had independently discovered Mendel's law of recombination. Chermak doesn't count because he wrote his own plaque before he died, which said that he'd done it. So, and of course, talking here, I've got to mention Bateson as the promoter of Mendel, but who had started the experiments that would probably have found the ratios before the fictional rediscovery on the train. That wasn't how it happened, but anyway. So, who invented Mendel's laws? So we say that the laws of the law of segregation and then the law of independent assortment, first and second law, weren't in Mendel's paper. Now there was many papers that appeared after 1900 that referred to Mendel's laws of alternative inheritance, etc. But none of them had none of these early papers had the first and second law. Most of them thought of dominance as possibly the primary law, for example, even though Mendel was clear that dominance wasn't an essential character of the actual plants. Dominance was necessary for him actually to be able to demonstrate segregation and independent assortment. If you had the many traits which blended, of course, you wouldn't be able to do that. So it was methodologically essential. Without that, he wouldn't have been able to actually separate out the trait from the factors that the plants had. Spillman, say in 1901, published his paper, and by 1902 Spillman was writing a paper on exceptions to Mendel's law, and you'll see here that he says that he'd written this paper. He wasn't aware that others had published anything on the same subject. Which I think is possibly true. And then he must have thought he was damned because he'd then basically identified what we now call as linkage, and he was presenting this paper in Science in 1902. And at the end of that, he had to put a little footnote. Since the above was written, I find that Professor Bateson has proposed the same hypothesis. So he was pipped twice for this. William Causal, many people said this. R.H. Locke, I'll come back to. Obviously, speaking here, I have to talk about Biffin, who wrote a paper which I will come back to in 1905 on Mendel's laws, but these didn't contain Mendel's laws as we now think of them. The first person to do this was not Morgan, it was Robert Heath Locke, in an utterly remarkable book. It's available online now. It's an extraordinarily advanced text on heredity, variation, and evolution. In it, he used the term Mendelize, which he said was a phrase coined by the Germans, meaning these random processes that are in law 1 and 2. That could be given a verb form of Mendelize. Mendelize was actually not introduced by a German. It was introduced by the amazing very interesting Liberty Hyde Bailey in a paper in Science in 1903. And Bailey is one of the few people who before 1900 had referenced Mendel several times. And de Vries actually wrote to Bailey saying, I came across the reference in the 1896 or 7, but from you, etc. But he later said he hadn't. But anyway, it's a very interesting story. Locke, however, formulated, and in the order that we know them, the first and second law, so the complete segregation of the two allelomorphs in equal numbers of the germ cells. That's the first and most important section of the generalization known as Mendel's law. And the second part of the law refers to the fact that, as a general rule, separate pairs of allelomorphs segregate quite independently of one another. So that is 1 and 2 in the way that it's now mainly taught in outside of Germany as being Mendel's laws. And he also noted the exceptions that Spillman noted and identified linkage. The laws are usually put down to Thomas Hunt Morgan, and the first time they actually appeared as 1 and 2 was in not very well known but interesting book in 1916, Critique of the Theory of Evolution. It's often attributed to the 1915 mechanism of Mendelian heredity, but it's actually not in the first edition. It was only in the second edition of that. And two of the co-authors of that, Hermann Muller and Alfred Sturtevant, said that Morgan gave them Locke's book to read. Everyone at the lab was given Locke's book to read when they turned up, but Locke was not referenced. Wasn't referenced for providing any actual intellectual input in Morgan's book. I think Morgan's a very sympathetic character, but he had reason to want to have some priority in the Mendelian field because he had been vigorously both anti-Mendelian and he'd been vigorously anti-Darwinian until 1910, and then sort of was converted to be the strongest supporter of both. So he had reason to want to appear to have been contributing to this area. So so this is just him. This is just Thomas Hunt Morgan being critical of Mendel. So moving on to Mendel's pure chance, as I say, I think we can see this as basically the combinatoric completeness, and this is what the first textbook, a proper textbook of genetics, in a way that we'd now recognize a textbook of genetics in 1925, talking about segregation and the theory of probability, said the surest evidence that chance alone is concerned in determining the combinations between different types of gametes is the similarity between the results obtained in the F2 ratios and those which should theoretically appear on the basis of probability, as illustrated by such a familiar example as coin tossing. And just in case you weren't familiar enough with it, they gave you a picture of what coin tossing was. Or in Lancelot Hogben's brilliant, honestly, it's a book I certainly learned a lot from reading relatively recently his Introduction to Mathematical Genetics in 1946. Hogben actually gives a definition of randomness based on explaining, picking cards from a replacement pack, saying that if the frequency with which we draw a particular card or one card of a particular class approaches a limiting figure identical with its proportionate possibility after an indefinitely large number of trials, our choice is random. So that's a nice definition of what random is, and obviously would then label Mendel's laws as laws that were related to randomness, which is how they are often presented in current textbooks. Now, Arthur Duckenfield, Derbyshire, was a very interesting case of an anti-Mendelian or biometrician-related researcher who converted to Mendelism. He wrote this wonderful book, Reading in the Mendelian Discovery, highly recommended as a read, in which he told people how to do the crosses, and actually gave them an address where they could write and get the seven trait varieties of Mendel's, and they could repeat the experiments and see what they saw. And he ran very large-scale studies himself, but he was summarizing the data of others, noting that the proportions lay very close to the expectation the expectations on the basis of pure randomness and probability. And the chromosomes became part of the evidence, if you like, for Mendelism, and Mendelism became part of the support for chromosomes. And Walter Sutton, who first put forward the notion that chromosomal division could give a mechanistic understanding of the Mendelian laws. It was very clear that about the role of chance in their separation. Even though he was completely wrong, and they thought that the chromosomes, the maternal and paternal, joined end to end, and etc. So it's interesting that what he was seeing when he was seeing this pure chance happening is difficult now to work out. And Eleanor Carruthers, who in 1913 looked at the situation with the sex chromosomes, again talking about chance, and that this provides support of Mendel's laws. Again, only thought this applied to the sex chromosomes, but the two came together. Really, each supported each other, and then of course the actual development of the understanding, the crossovers during meiosis occurred. And Locke in 1906 was already saying, had a whole chapter on cytological evidence for Mendelian inheritance. So what do these random processes provide us with? Well, I mean, they provide the null model for population genetics and beyond, usually the Hardy-Weinberg equilibrium put forward in 1908, formally being that null model. That's what you look at as with expectation, and its deviations from that would say there's selection or other processes. As an epidemiologist who often does genetic epidemiological approaches, many of the genetic epidemiological approaches simply use the Mendelian expectations, again, as a counterfactual, as your null in the transmission disequilibrium test, for example, or just in a straightforward population case control study. So that's what's used as the null. So, and of course, Markov didn't himself define his processes. The notion that something purely depends upon the one state before, that prior to that nothing has an effect, which allows mathematical, relatively straightforward mathematical solutions to problems. So you don't need to go back beyond the single state before. Retrospectively, Mendelian processes are seen as Markov processes. And this, of course, is seen most clearly in the relationship to the Mendelian view of heredity than the law of ancestral heredity of Galton, where where half of your phenotype influenced by your genotype came from your parents, and then a half came from previous generations, i.e. a quarter from your grandparents and eight from your great-grandparents, etc. That was the very obviously non-Markovian process that underlies the law of ancestral heredity. Derbyshire, as always, in his book, rather beautifully illustrates this by saying, your expectation here would not be, if you thought of in the terms of the law of ancestral heredity, would not be the same as your expectation here. But what you actually get here, of course, is when you actually do the hybrid selfing, would be 75% yellow, 25% green. So it's a lovely book presented in an educational way, and notions are given that, for example, Haldane's models were very much Markov models, before Markov. So people rather anachronistically refer to hidden Markov, Haldane's hidden Markov model, when Markov hadn't identified those processes. The Mendelian randomness also allows for directly estimated heritability, where if you have, for example, sibling pairs, then you have your mere chance is that they'll have 50-50 sharing when you look, but of course they don't have 50-50 sharing there is a distribution around the amount of sharing because of the unequal crossovers. And so what you can do is just regress the phenotype similarity in SIB pairs against the exact sharing, which gives you an estimate of heritability, which doesn't have all the problems of twin heritability and arm monozygotic twins brought up in a similar way as dizygotic twins, etc. So the Mendelian randomness, together with knowledge of meiosis and crossover, allows you to estimate that. And another possible importance is with respect to preventing genetic cheating. There's an interesting book by the evolutionary biologist, but mainly known as the textbook writer of what used to be the most read textbook of evolution for decades, Mark Ridley, a book he wrote 20 years ago called Mendel's Demon, Gene, Justice and the Complexity of Life, which got reviewed in Nature and Science. It got endorsed by many very well-known people, but seemed to sort of disappear, but just has this notion that it is the randomness which just prevents genetic cheating. Without those random processes, then segregation distorters, for example, would take over, and complex life wouldn't be allowed to exist. And there's an analogy here with the moral philosopher John Rawls's notion about the veil of ignorance. So the veil of ignorance would be in a society. So now moving outside of Mendelian genetics, but to come back very quickly to it, would be if you were someone who was going to choose the rules of society, and you were going to choose those rules in a situation where you didn't know what your location in society was, you would tend to choose rules that would be fair. Whereas once you are in a position where you know that you're in a privileged position, you manage to justify rules which are unfair, and you manage to convince yourself that it's your wonderful abilities which have got you into this position, rather than luck, exploitation of others, etc. So there's maybe an analogy with the fair meiosis. Has the genes in the same position. They don't know whether the randomness means they don't know where they're going to go, and if they're going to go through to the egg, and if they're going to go with other partners they're with, etc. So maybe those processes do allow for complex life. Now, I mean, there's vast literature on the evolution of meiosis, and many theories about why it evolved, but probably which and no agreement on them. But this is one possibility: is that by preventing genetic cheating, they actually allow the development of complex life. And of course, meiosis also provides for selection. It provides fair tests for each variant, each genetic variant or mutation, because it will be against backgrounds which are just randomized to it. So every mutation will, in the end, sort of be tested against equal probability of getting particular backgrounds. So it provides the optimal mechanism for sort of successful selection. So those are the randomness in Mendelian processes in a biological sense, if you like. And I'm going to move on now to talk about Mendelian processes and causal inference, of which Derbyshire said, "At this point, therefore, we bid farewell to the broad daylight of observation and enter the dark and treacherous alleys of inference." So I'm not going to talk about R.A. Fisher's claims that Mendel's data were faked, that the mere chance was too little, indeed, that there were in them. I think there's been enough discussion of that, and it's likely that things that weren't understood at the time in experiments, like stopping rules that you have to say in advance when you're actually going to stop counting the data, weren't thought of, and so there'll be multiple points of stopping, and you just stop when it looks close to your belief. Doesn't mean that it's fraudulent. I said I wasn't going to talk about it, but I said I did a bit. But I thought, being here, I had to mention the first. This is the first Bateson. This is the first Bateson lecture delivered here. So I thought I had to discuss Fisher's statistical methods in genetics paper, because this is the one time in his career Fisher spoke to both geneticists and statisticians. I liked it enough to reprint it in the International Journal of Epidemiology when I was editor of it many years ago. And in this, Fisher basically talks about Mendelian randomization. He says, genetics is indeed in a peculiarly favored condition in that providence with a capital P has shielded the geneticist from many of the difficulties of a reliably controlled comparison. The different genotypes possible from the same mating have been beautifully randomized by the meiotic process. Generally speaking, the geneticist, even if he foolishly wanted to, and it was a he, of course, could not introduce systematic errors into the comparison of genotypes, because for most of the relevant time, he has not recognized them. So this is beautiful randomization. And he said a connection between our two subjects, which seems not to be altogether accidental, namely that the factorial method of experimentation, that's when you randomize two different treatments factorially in plants, of course, which is Rothamsted for a long time, now of lively concern, so far afield as for psychologists or the industrial chemists, derives its structure and its name from the simultaneous inheritance of Mendelian factors. Geneticists certainly need not feel that the intellectual debt is all on one side. And Fisher's daughter, one of Fisher's daughters, Joan Fisher Box, who married to the statistician of the time, George Box, I emailed her when I reprinted Fisher's paper, and in her 90s, she wrote an absolutely brilliant commentary on the Bateson lecture. And she said that that Fisher got his notion of randomized trials. So I'm not sure people here, Fisher is considered to be the person who introduced randomization, randomized experimentation in large-scale studies, in agricultural studies. That his notion for randomized trials came from factorial randomization by Mendel's laws. So Mendelian randomization came before randomized controlled trials. That's the lesson. Fisher got that model from that. And if you read Fisher's early papers on randomization, it wasn't about balancing confounders, etc. It was about getting correct variance estimation. Now, one of the other utterly brilliant contributors to genetics and evolutionary theory is Sewell Wright. Wright, I always loved this in the National Academy of Science biography of him. He read his father's math books and learnt to extract cube roots before entering school, a skill that he said brought him instant, lasting popularity with the other students, something I'm sure some people here can recognize. And Wright introduced a form of causal analysis that he referred to as path analysis. Wright introduced this purely in genetics because you have the anchors from the genetics, but of course in his case it wasn't molecular genetics, it was doing crosses. And this is his famous illustration of his path analysis looking at guinea pig coat color. And what I love here is he actually just drew chance, and this is just chance going with the gametes and fertilization. So he saw chance as being which underlay these processes, meaning that you were getting essentially experimental data, getting an experiment that you weren't conducting, but you were using a natural experiment. And he did some very high level causal inference. So, for example, calculating that in just your sort of standard guinea pig crosses, 58% of the variance was what he called intangible. I think this is something which is just forgotten. We all, have this dream of personalized medicine and complete explanation. But as Wright pointed out, in his totally controlled situations, you get 58% of the variance must be due to irregularities of development, due to the intangible sort of causes to which the word chance is applied. And he also showed that, of course, if you had highly inbred guinea pigs, then the intangible variance was 90% or 80% or something. But and that was, and so a big chunk of the variance was caused by stochastic processes. And he carried out a whole stack of analyses using path analysis, but in all of them, that you had a very strong causal anchor, almost always genetic. You didn't just you couldn't just apply path analysis to any cross-sectional or whatever prospective observational data you had. But path analysis became really widely used in the social sciences. Wright would have been, I think, upset by this, because he said repeatedly, he said that the only reason you could make causal statements was because of the Mendelian processes. And he was definitely upset by the way people were using path analysis and making claims without having the causal analysis. And the statistician Louis Gutmann, back in 1977, sort of satirized the situation in the sort of social and psychological sciences, producing these path analyses, saying that genetics has but one modest framework for paths. In contrast, according to the current journals, sociologists keep discovering new fundamental path frameworks every month, and sociological graduate students are required routinely to hand in as individual class exercises new discoveries equaling Gregor Mendel's. He didn't think this was the case. He thought they were just producing bullshit, but making causal claims from it on the basis of the analysis. So using molecular genetics as an anchor for causal inference is what Mendelian randomization does. It's 20 years since been promoting this, and I'm still around flogging it 22 decades on. But the question is, why would you need to use genetic variation? Why not just measure things and see how they related and statistically adjust, etc.? Well, I mean, the thing is that in the end of the last century, there was just lots of nasty surprises in epidemiology. Here's a couple of papers in the New England Journal of Medicine looking at people taking vitamin E supplements and the risk of coronary heart disease. And you saw in one large cohort, there's about 40% lower risk of coronary heart disease in people who were taking vitamin E supplements. This was replicated in another large cohort. It was the headline in the New York Times, moving Clinton to the second headline. Vitamin E greatly reduces risk of heart disease, studies suggest. That's obviously a causal claim, greatly reduce the risk of heart disease. Vitamin E supplement use zoomed up in the US till at one time over about 40% of adults were taking a supplement containing vitamin E. And then, of course, the randomized controlled trials appeared, and they showed absolutely no effect of actually randomizing people to vitamin E supplements. But you were studying precisely the same exposure. And this happened again and again and again with different factors. That epidemiology was identifying these factors as being potential causes, and randomized trials were showing this to be spurious. Indeed, there was this cartoon appeared saying, today's random medical news from the New England Journal of Panic-Inducing Gobbledygook, and the news reader just presses the button, and according to a report released today, and these sort of spin, coffee causes depression in twins, etc. And then you just press the button and you read out the next one. And every one of these is, of course, spurious, like stress doesn't cause breast cancer in 7 out of 10 women. The only one which is true is that red wine does cause sexual dysfunction in men aged 25 to 40, but it's a very long time ago that I have causal evidence on that. So conventional observational epidemiology is you just measure your exposure, vitamin E supplementation, whatever. An example would be C-reactive protein. C-reactive protein is an acute phase reactant. Everything bad that happens, you get an infection, you smoke a lot, etc., increases your CRP. But this reverse causation, so like early stages of coronary heart disease, atherosclerosis is an inflammatory process, bangs up your CRP. You've got confounders, smoking, social class, etc. You can't deal with those statistically. But if you have a genetic variant, like a SNP in the promoter region of the CRP gene, which is related to C-reactive protein, associates reliably with that, then of course there can't be any reverse causation and there won't be conventional confounding. So your joint association of CRP, the CRP variant and CHD, and CRP variant and CRP allows you to estimate the causal effect of CRP on coronary heart disease. So the fundamental assumption, if you're going to use this for talking about interventions, is of gene environment equivalence, i.e. the genetic influence on CRP. If the component of variants of CRP influenced by genetics will have the same effect on the disease outcome as by environment. And there's sort of examples of this equivalence, such as Hartnupp's disease, a monogenic disorder, looks very much like pellagra, which is caused by niacin deficiency. And the Hartnupp's disease, the genetic process leads to lower niacin. So you have to make this assumption that the effect will be the same, independent of whether it's genetic or external influence. So that's the gene environment equivalence. I mentioned CRP and coronary heart disease. C-reactive protein was a very strong predictor of coronary heart disease. It was called the new cholesterol. Drug companies were introducing agents, were studying agents that would lower CRP. You see that your multivariable adjustment leaves quite a large, very robust effect, just like with vitamin E and coronary heart disease, for example. But if you look at the confounders, as I said, CRP, your measured CRP just relates to everything that's bad. But if you look at the genetic variant related to CRP, it just doesn't relate to any of those confounders. So Mendelian randomization does what it says on the tin. And your estimate of causal effect of CRP is flat. Drug companies stopped developing CRP lowering agents. Now, MR was introduced to strictly Mendelian processes, i.e. as something that was within families or between siblings. So it was the randomization of the alleles from parents to offspring. And that it would be analogous to an RCT. But there just weren't large, there were just no family studies back then. And so we suggested that Mendelian randomization and genetic association studies is approximate rather than absolute, but you could use these principles in observational data, just straightforward population data if you didn't have SIB pairs, and there was approximate randomization. But now there are large enough. We've got a consortium of 100,000 SIB pairs, so Mendelian randomization can be carried out in SIB pairs. I said that it's like an instrumental variable approach, and instrumental variables was interestingly introduced by Sewell Wright and his father, but not applied to genetics. And of course, and T.H. Morgan, in his Nobel Prize acceptance speech, talked about how geneticists just use genes to tell them about downstream processes. He was very clear about this. He wasn't really that bothered about what was in the gene. It was just used to tell you about phenotypic causality. So an instrumental variable just has to be associated with your exposure. There has to be no confounding of the instrument and outcome, and there has to be here's an example of a parent offspring form of analysis where if you're looking at where the mothers have a genetic variant that relates to lower folate activity, which we know now from randomized trials increases neural tube defects, then the offspring has a twofold risk of neural tube defect. If the father carries that variant, there's no effect because it's an intrauterine influence. And if the offspring carries that variant, then they show an increased risk, but that increased risk is due to the maternal genes. So that would be a sort of confounder, would be the maternal genes in this situation, would be a confounder. But if you understand the mechanism, it gives you good evidence about the effect. And then there has to be no horizontal pleiotropy, meaning that there must be no effect of the gene on the outcome except for your exposure of interest. Now, there's two forms of pleiotropy. There's vertical pleiotropy, where a genetic variant influences an exposure, and that influences a downstream factor. Mendelian randomization is just vertical, it's just utilizing vertical pleiotropy. But it's horizontal pleiotropy. Everything, of course, has to be through a phenotype, so the gene doesn't directly affect your outcome. It's horizontal pleiotropy which causes problems for Mendelian randomization. And there's a vast industry producing methods that do allow testing of horizontal pleiotropy. Mendelian randomization can be carried out for more than one exposure when you have genetic instruments which relate, which can then be related to both exposures and outcomes and assess the effects. And sort of complex form of multivariable MR will be on childhood and adulthood body size and their effect on outcomes. There's been a lot of interest are there independent effects, residual effects of greater childhood BMI, for example, on outcomes. You can look at proof of principle examples showing that the method of multivariable MR will separate out childhood and adulthood BMI and the effects on childhood vitamin D. Your adulthood BMI can't influence your vitamin D in childhood, obviously. So this is quite a difficult positive control test, but the multivariable MR does indeed show that. So that genetic variance, your childhood body size influences your childhood vitamin D, but there's no effect of adulthood body size on childhood vitamin D, which observationally, of course, you get very strong effects, and you can't get that out of observational analyses. And similarly, in the multivariable MR, childhood BMI does relate to adulthood vitamin D, but of course, childhood BMI influences adulthood BMI. So when you take that into account, there's no remaining residual effect of childhood BMI on adulthood vitamin D levels. And another proof of principle, type 1 diabetes generally seen in children, that childhood in the multivariable MR situation, you see that childhood BMI increases the risk of type 1 diabetes. Adulthood BMI in the univariable situation appears to relate, but in the multivariable doesn't. You get totally null effects. The reverse is true for type 2 diabetes. There's no residual effect of childhood BMI. It does associate with it, but there's no actual effect if you take into account adulthood BMI. So these are things that you just simply can't analyze in observational studies. So I'm just going to finish with a ridiculous claim that natural selection is a Mendelian randomization problem, because if you think of your phenotype not being type 2 diabetes or whatever, if you think of your phenotype as fitness, and of course, selection works through phenotype. It doesn't work through the genes aren't selected for. It's the phenotype that is influenced that's selected for. Your genetic variants can be used to work out what actual factor is being selected for. Gene environment equivalence applies in selection. I mean, I'm sure many people here have used the breeder's equation. If you for the breeder's equation, if you use this, you are making a gene environment equivalence assumption. You look at your selection response. That's how much you gain when you make matings from selected parents. That's your selection response is the heritability, multiplied by your selection differential. The selection differential is the difference of the base population mean and the mean of the selected parents that you're actually using in this selection. And your heritability, of course, is your genetic variance divided by the total phenotypic variance. So this equation means you're assuming that the genetic component and the non-genetic component of the phenotype have exactly the same, have exactly the same selection effects. But you can use the genotype to work out the full phenotypic effect. But of course, in the selection case, you're interested in the genetic equation. So you could, pleiotropy is an obvious issue in selection. famous farm fox experiment, you know, when these tame foxes were kept for 70 years and bred and bred and bred and bred and bred, and they became very tame, and they also got huge bushy tails. Can you separate out which thing has been artificially selected on if you actually had genotypes in those creatures? And these approaches over the last couple of years, this was the first one I saw, but now there's quite a few papers that are applying Mendelian randomization using genetics in, for example, rice. So natural selection is just a subcategory of Mendelian randomization, and randomized trials just mimic Mendelian randomization as well. So that's the punchline. I'm going to finish with 1 minute coda, if I'm allowed to. So I mentioned Biffin. Here's Biffin's paper again. I'm sure everyone here has read Biffin's paper, and you'll all know that he said, in this country, the work of the Gartons, which has resulted in the introduction of a number of fresh varieties characterized on the whole by high-yielding properties, is the best known example of selection. Little beyond popular descriptions of their work has, however, been published. And here is John Garton, and this is one of their seed bills from, you could buy all these wonderful seeds. And you'll notice Garton's Limited of Warrington. So I'm interested in this because I come from Warrington. This is an advert for Warrington, the worst town for culture in Britain. There was a survey done by the Royal Society of Arts. Warrington came 355th out of 355 as having the lowest level of culture in Britain. And then the government did a quality of life survey or scale using routine data, and Warrington came bottom of the list of quality of life. So you've got to realize how much Biffin and Biffin quoting Gartons means to me, because it might be fucked culture-wise, but it is the home of aristocratic seeds. There you go. And the other extraordinary thing from Gartons were, this is another one of the adverts, some of you might know about this, is their intellectual property donation. The Gartons tried, in 1899, the Gartons tried to donate their seeds to the government as an intellectual property donation, which was turned down. So generous, as well as aristocratic, generous seeds. And if anyone's interested in Mendelian randomization, there's a very recent Nature Reviews Methods primer, which gives everything you need to know about Mendelian randomization. And if you're looking for Christmas presents, I can highly recommend this book as an ideal present for anyone you really don't like. Okay, thanks very much.
Thank you very much, George, for a very interesting and entertaining talk. So as per usual, we'll ask for questions and ask for questions from postdocs and students first. This is an open lecture, so we'll have some questions coming in on Zoom, perhaps. We'll be looking for those. So now is the time to raise your hand. This has been food for thought. Any questions from the audience? And we have two runners here who are going to come and give you a microphone. It always takes a while. It takes a while. It takes a while for people to cogitate. So we'll just be patient and let those cogs turn. And do we have anything on the? All rolling. Comments? Shall we? Right, an interim question from a non-student.
Student of life, surely.
So thanks, George, for that tour de force. And a number of things sort of come to mind. One is this business about mere chance, and Darwin also uses the term mere chance. I've forgotten the quote exactly, but it's something along the lines that if this were due to mere chance, all this would be meaningless. So he thought that mere chance was not involved in the Darwinian evolution. So was he using mere chance in the sense that you use, or was he really meaning pure chance? Would you like to tease that one out?
Yeah, that's really interesting. Yeah, Darwin was clear that he thought he had the sort of ignorance view of what we put down to chance, i.e., saying something was due to chance reflected ignorance of some mechanism that must exist, but that we can't understand. And I think by that, Darwin's is very confusing in the way he used the terms fluctuations. He definitely used the word fluctuations in different ways, and his followers use the term fluctuations in different ways. And that people usually think of fluctuations as a thing which is noise, is mere chance. But for Darwin, the fluctuations, those small differences, were the things which were heritable. Of course, that evolution was through small changes, not through saltations, not through large leaps. So I think Darwin was using mere chance. He was thinking about things which were just measurement error, error, etc., which obviously couldn't drag. Obviously the mechanism of natural selection wasn't known, and we could go into an interesting discussion of Fleming-Jenkin and how Mendel helped with that problem. But was that if these things were just chance, then you couldn't drag the phenotype with it. It couldn't actually lead to selection. That there had to be some connection that would allow those things to be dragged along.
We have one, yes, Parita.
Thank you for the interesting talk. So I was wondering about the example you gave about the vitamin E and it being not having an effect. That was in the 90s. How has the research and the community come forward since 90s? So are there still these papers all the time saying this is what happens when it's just mere chance or the variability? Or has the medical field matured after that and everyone is expecting randomized Mendelian trials?
No, I mean, I think if you follow the sort of health scare headlines, then studies that are observational studies where confounding reverse causation might well be generating the findings, get widely reported. I mean, there was one, a few days ago suggesting that it was better, the lowest mortality rate is in people who are a bit overweight. That was the group who had the lowest mortality. But it's definitely, there's situations, there's a lot of confounding, a lot of reverse causation. Mendelian randomization suggests rather like the very sad result with alcohol when there's meant again papers come out again. There was a relatively recent one suggesting that moderate alcohol consumption reduced heart disease risk, for example. Sadly not true. Well, there's not been a randomized trial, but the Mendelian randomization, for which in the alcohol situation they have very powerful data, because in East Asian populations, there's a genetic variant that relates really strongly to alcohol consumption. So you can define groups that have different levels of alcohol consumption by their genetic variants, which is what Mendelian randomization does, and do an intention to treat analysis, thereby shows no benefit, just consistently adverse effects in men. And then you have the wonderful negative control of women in those East Asian populations hardly drink. So if it was something else to do with the gene, the genetic variant, if it wasn't due to the alcohol consumption, you would see the same associations in women. But amongst women, there's no association of the genetic variant with cardiovascular disease. So, but yeah, no, the bottom line is the stuff, the nonsense still appears. And sadly now, some of them not. There's a lot of very, very bad Mendelian randomization papers. So it's too easy to do. So that doesn't mean that the answer is going to be right. I don't think in causal inference. I think it's very rare where the best approach is just to use one method and trust that. The best approach is to consider different ways of estimating causal effects, different methods which have different assumptions. And it's when the different methods with different assumptions and where the biases, everything can be biased, but where the biases wouldn't be the same in one study design as in another study design. It's when those things come together in a sort of triangulation of evidence framework that you can have a stronger belief in causal effects.
Yeah.
And
Yeah.
you talked about Mendelian randomization and properties of meiosis, but as you know, there are a fairly large number of people here who work with prokaryotic organisms that don't go through meiosis, and I think you could argue fairly convincingly that more or less the vast bulk of biological diversity is in prokaryotic organisms. So how does that fit with your ideas about Mendelian randomization as being responsible for, I suppose, biological diversity in a loose sense?
Yeah. So I think the notion of the requirement for meiosis is for forms of more complex life, and there's many more things can break down. I mean, as you know, the work on the minimum genome, I think it's like 200. I can't remember how many it was. I only got this from a podcast. So, but you know, it's this sort of 250 genes. But even in that situation, you can mutate one by one, and there's you know, incredible robustness. But I think rather like with Mendel having the very large numbers, you actually quantity turns into quality. I.e., you know, just simply amassing more data, you get answers that you believe. That's one example of quantity turning into quality. I think with biological organization, as things get more complex, there's just many more things that can go wrong. I mean, you could say that you can have me alive without meiosis, but there's a limit on the complexity that can be developed. Because I mean, we've got to remember that all we know is basically what we know is what's around, and so it's almost a circular argument of saying, if we say what's around, and then we say there must be reasons for that. There might be lots of reasons for why there aren't, you know, transitional forms, or the complexity is limited.
Okay, I think we're going to wrap it now because it's past,
Sorry.
well past 12:30. So George, we're letting you off the hook. Thanks once again.
Thank you.